Good morning everyone. Today we are going to discuss about the various biological processes that are involved in drug targeting. Basically, when we talk about drug targeting, it indicates the delivery systems or the drugs, what is their efficiency to reach their respective destination. These respective destinations can be either the tissues, organs or the cells or the intracellular organelles present inside the cells. Depending upon the various receptors that are attached onto the tissues or onto the cells, the systems, either the drug or the delivery systems, they can be recognized very easily. Besides this recognition by the cells or the tissues, the other biological processes that are involved in drug targeting will be discussing in this particular presentation. Now, oral administration of therapeutic agent is a preferred means of delivering drugs from the standpoint of ease of administration and patient acceptability. Formulating a drug for oral delivery is more often than not a complicated process. However, methodologies for systematically optimizing a drug's stability in gastrointestinal tract, obtaining the desired pharmacokinetic profile and biodynamics, as well as reducing absorption variabilities and side effects are not well established, routine or trivial endeavors. Delivery issues become even more complex when the pharmaceutical scientist is confronted with designing formulations for the ever expanding number of protein and peptide therapeutic drug candidates that are currently being identified and evaluated for oral administration. In this presentation, we will be discussing about the following points that are the various biological processes that are that you can see on the screen, like cellular uptake processing, the transport across the epithelial barrier, extravasation, lymphatic uptake and function of reticuloendothelial system. So let us start with the first process, that is cellular uptake process. The cell membrane employs different mechanisms to exchange substances which are mainly divided into two categories, that is passive transport and active transport. Gases like oxygen and carbon dioxide, hydrophobic molecules like benzene or uncharged molecules such as water and ethanol easily diffuses across the membrane from the regions of higher to lower concentration. This kind of transport, which is along the concentration gradient and occurs without any assistance of energy, is called as the passive transport. So the passive transport is basically the normal transport process. In contrast, the active transport occurs against the concentration gradient by using the energy which is provided by the ATP, that is adenosine triphosphate. The polar or uncharged biomolecules that cannot pass through the hydrophobic plasma membrane are internalized by a form of active transport which is called as the endocytosis. In this process, the cell engulfs, that is in the endocytosis process, the cell engulfs the materials inside the extracellular fluid by invagination of the cell membrane and buds off inside the cell, forming a membrane bounded vesicle which is known as the endosome. Now, this endocytosis process can be basically classified into two major categories, that is phagocytosis and Pinocytosis. In case of phagocytosis, which is known as the cell eating, it is the process of taking in debris, bacteria or other large sized solutes by specialized mammalian cells which are called as the phagocytes. These phagocytes are basically monocytes or macrophages as well as neutrophils. Integral to the phagocytosis is a process called as the opsonization, by which opsonins like immunoglobulins and complement protein coats the target materials to trigger the phagocytes of their presence and to localize the phagocytic activity. As the phagocyte begins to ingest the target material, it will simultaneously stimulate the formation of membrane-bound vesicles called as the phagosome, into which the ingested materials are compartmentalized within the phagocytes. Now, this phagocytosis basically at a later stage of the phagocytosis process, the phagosomes will fuse with the lysosome and the material will be digested by the acidic pH present at the cell by the hydrolytic enzymes contained within the lysosomal lumen. In case of pinocytosis, in all the cell types, small particles within the range of nanometers are internalized by the pinocytosis mechanism. In this process, that is the cell drinking mechanism, plasma membrane forms invagination to take up a small droplet of extracellular fluid, including the dissolved molecules in it. Pinocytosis is not a discriminating process and it occurs in almost all the cells in a continuous manner, irrespective to the needs of the cell. The grabbed substance are pinched off into the small vesicles that are called as the pinosomes. The pinosomes are basically the small vesicles which grabs the substances of smaller particle size within and which within the cells, which fuses with the lysosomes to hydrolyze or break down the contents thereby. The phagocytosis and pinocytosis can be distinguished by their size of their endocytic vesicles. In case of phagocytosis, there is an uptake of large particles by large vesicles within the size of 250 nanometer range, and in case of pinocytosis, there is uptake of fluids through small vesicles with the size range of few nanometers to 100 nanometers only. The pinocytosis can be subcategorized into Clathrin-mediated endocytosis, Caveolae-mediated endocytosis, Clathrin and Caveolae-independent endocytosis, and Macropinocytosis. In case of Clathrin-mediated endocytosis, this is basically a cellular entry mechanism to internalize specific molecules into the cells. This entry route aids the cells to take in the plasma membrane components and nutrients, including cholesterol, by low density lipoprotein receptors and iron by transferrin receptors. In this process of Clathrin-mediated endocytosis, particular ligands in the extracellular fluid bind to the receptors on the surface of the cell membrane, forming a ligand receptor complex. This ligand receptor complex moves to a specialized region of the cell membrane, which are rich in clathrin, whereby they are engulfed through the formation of clathrin coated vesicles. Once inside the cell, the clathrin coatings on the exterior of the vesicles are expelled prior to fusing with the early endosomes. The cargo within the early endosomes will eventually reach the lysosome via the endolysosomal pathway. In case of the Caveolae-mediated endocytosis, this is the route of cellular entry which involves flask-shaped membrane invaginations called as the caveolae, little caves. The caveolae are present in the endothelial cells, epithelial cells, adipocytes, muscles, and fibroblast cells. The size of the caveolae typically ranges from 50 to 80 nanometers only and are composed of membrane protein, that is caveolin 1, which gives them the flask shaped structure. The caveolae dependent endocytosis is involved in cell signaling and regulation of membrane proteins, lipids, and fatty acids. Once caveolae are detached from the plasma membrane, they fuse with a cell compartment called as the caveosomes that exist at a neutral pH. Caveosomes are able to bypass lysosomes and therefore protect the contents from hydrolytic enzymes and lysosomal degradation. Hence, pathogens including virus, bacteria use this entry route to prevent their degradation. Since the cargo internalized into the cells by caveolin dependent mechanism do not end up in the lysosome, this pathway is basically employed in case of nanomedicines. In case of the third mechanism, that is the Clathrin and Caveolae independent endocytosis, this basically occurs in cells that are deprived of Clathrin and Caveolin. This pathway is utilized by the growth hormones, extracellular fluid, that is ECF, glycosyl phosphatidyl linositol, that is GPI, linked proteins and interleukins 2 to enter the cells. For instance, folic acid that employ Clathrin and Caveolae independent pathway to enter the cells are conjugated to the nanoparticles and the polymers used in the drug delivery systems as an imaging agent. Macropinocytosis is another mechanism. This is a type of pinocytosis mechanism in which the cells take in high volume of extracellular fluid by forming a large vesicle of size 0.5 to 10 micrometer called as the macropinosomes. Macropinocytosis is a pathway to internalize apoptotic and necrotic cells, bacteria and viruses as well as antigen presentation. This pathway can internalize micron-sized nanoparticles which are not possible to be taken up by the cells by most other pathways. Macropinocytosis mechanism can occur in almost any cell except for the brain, microvessels, endothelial cells. This macropinocytosis is a mechanism to internalize apoptotic and necrotic cells. Already I have told you. In case of the fourth mechanism, that is transport across the epithelial bed. Next mechanism, first we have discussed about the cellular uptake process. So, in case of cellular uptake process, we will just have a roundup. The cellular uptake process basically it is composed of the pinocytosis as well as the phagocytosis, and the pinocytosis and phagocytosis, there are particular differences between the pinocytosis and phagocytosis, as well as the pinocytosis is governed or it is subcategorized into certain mechanisms, that is your Clathrin-mediated endocytosis, Caveolae-mediated endocytosis, Clathrin and Caveolae independent endocytosis and Macropinocytosis. In the next presentation, we will be going through the next biological process, that is transport across epithelial barrier. Till then, take care. Thank you.